Hello students, welcome back to our course Environmental Modeling and Simulation. In today's class, we are going to apply all the principles that we have studied so far on how to build a model, and we are going to apply it for a very important environmental challenge, which is spread of antibiotic resistance in environmental matrices. So I'm going to take some time to introduce to you what the problem is and how the empirical approach can be utilized to model the spread of antibiotic resistance in one particular environmental container. So let's get started. This is the paper that we are going to study today, a model to investigate the impact of farm practice on antimicrobial resistance in UK dairy farms. The principles applied in this particular paper are exactly what we have studied so far in the course, and what we are going to and this example is also relevant for other environmental systems such as aeration tank and an ASP or other environmentally related processes such as wastewater treatment, WSPs and wastewater receiving ponds. Okay, so this is quite a recent paper. This is research that was completed about 2 years ago. And as I said, I'm going to introduce to you what antimicrobial resistance is. So let's get started. Antimicrobial resistance is the ability of microbes or bacteria to resist antibiotic, or in this case antimicrobial therapy. So the name itself is little counterintuitive. So just to be clear, antimicrobial resistance also referred to as antibiotic resistance when we are talking about just antibiotics, is the ability of bacteria to resist antimicrobial or antibiotic therapy. So if one bacteria that used to be sensitive to an antibiotic or antimicrobial now develops resistance against it, then that microbe is known to be antimicrobial resistant. How does this happen? How does it spread in environment? Very brief overview of the processes will help you understand how to write the empirical model for this. So the way a bacteria that used to be sensitive, SS was sensitive here, acquires antimicrobial or antibiotic resistance is either another resistant bacteria gives it genes that are required for antibiotic resistance. So certain genes that are specific to developing resistance. This is one way. Now the other way is that if in a population we have both sensitive and resistant bacteria, and now we are adding antimicrobials or antibiotics in this environment, then the sensitive bacteria will die out, leaving only the resistant bacteria, which will over time increase the number of resistant microbes. And what we will see is that over time we don't have any sensitive microbes left, or we don't have much sensitive microbes left, and most of the microbes that we have are resistant bacteria. So these are the two very overarching ways in which the resistant bacteria in environment increase. So to summarize, one is by gene transfer. The technical term is horizontal gene transfer. And the second is selection of resistant bacteria because of the pressure of antimicrobial in the environment. So we are going to include both of these in our empirical model. Now let's try to understand what particular environment this paper targeted. Now in UK dairy farms, the wastewater, the wash water that comes from the dairy farms, which will include the water used for cleaning, the water urine, and all other kind of wastewater that is produced in the dairy farm, is sent to a slurry tank. So let's look at the slurry tank. The actual volume of slurry's tank V remains constant over time, which is the case with most of the reactors that we construct. This slurry tank is modeled in this paper as a CSTR, continuously stirred tank reactor, which means that the concentration in the tank of the antibiotics, of the microbes, of the nutrients is homogeneously distributed throughout the reactor. However, the slurry tank does not act like a perfect CSTR. There is a slight difference, and the difference is that there is a phase in which the slurry tank is filled, So the volume, the effective volume of the slurry increases during that phase, and then after it reaches a particular volume or after a particular time, the slurry water is removed from the slurry tank. So there is a time period when the water is removed from the slurry tank. So the actual volume of the slurry tank keeps on changing. So the actual volume is variable and varies with time, and that's what we are going to include when we apply mass balance. Later in the course, I will teach you about other reactors that behave similarly, but right now our focus is on developing empirical models. So now let's go back to this point where we talked about how bacteria become antibiotic or antimicrobial resistant, and the two methods that I told you are horizontal gene transfer or selection pressure in the environment. How will we model that? Now let's think about gene transfer. So if you look at gene transfer, what I told you is that the resistant bacteria, here I am writing capital R to show resistant bacteria. What this resistant bacteria will do is it will give the gene that encodes for antibiotic resistance, antimicrobial resistance. There is a class of genes that people study which do this, which encode for antimicrobial resistance and can be transferred horizontally among the bacteria. And it will give this to a sensitive bacteria, which will now transform into a resistant bacteria. If you remember what we studied about autocatalysis and what we studied about other chemical kinetics in this course so far, I hope in your mind you're able to already see some parallels between what's happening here and what happens in autocatalysis. A resistant bacteria is interacting with a sensitive bacteria and converting it into a resistant bacteria. This is sort of autocatalysis happening, and if you remember that equation, we can write this like so. A resistant bacteria, a sensitive bacteria, they interact. Interaction is essential here, which should remind you that when two species need to interact for a chemical reaction to happen or for a phenomena to happen, in your model you're going to multiply. You're going to multiply their numbers to account for the interaction. This is something we discussed when we were talking about Lotka-Volterra models for predation, competition, and even cooperation. If in this case RNAs were cooperating, then and it was helping their growth rate, then dr by dt should include a term that has product of RNS. We also discussed this briefly in epidemic model when an infected person and a susceptible person had to interact for the contagious disease to spread. Here, a contagious disease is not spreading. Here, the only point of the interaction is is actual transfer, horizontal transfer of genes that will confer antimicrobial resistance to a sensitive bacteria. So what's happening here is a sensitive bacteria and a resistant bacteria are interacting with each other. There is a rate constant. So not all interactions between a resistant and sensitive bacteria will end up with the sensitive bacteria becoming resistant. So we are going to have a rate constant. But whenever the reaction is successful, we'll end up with two resistant bacteria for each interaction between a resistant and a sensitive bacteria. So let's say K is your rate constant. All right. Now, this model definitely uses this principle. So, in chemical kinetics, the way we understood this was that the rate at which dr by dt is changing with time is equal to KRS. There is no backward reaction in this case. The resistant bacteria we are assuming in this model will not turn into sensitive bacteria, which can happen under certain circumstances, but under the circumstance of this dairy this dairy farm, it has sufficient selection pressure that a resistant bacteria will not be interested, or it's unlikely that resistant bacteria will turn into a sensitive bacteria. So dr by dt is equal to KRS. Now we need to account for different kinds of resistance. So given that we are dealing with a dairy farm in this model, the authors of the study understand that there are different kinds of antimicrobials, and different kinds of antimicrobials require different genes, different methods through which the bacteria can develop resistance to it. So they classified the kind of antimicrobials into three different groups. Now, here I want to tell you something. It is possible to write a very simple model. Actually, this is a perfectly simple model, right? I just want to look at the numbers of resistant bacteria and sensitive bacteria, see how many times they interact. I need to find out this K, which will tell me at what rate the new resistant bacteria are being produced. But I'm missing a lot of information here. First thing I'm missing is that any antimicrobial, AM for antimicrobial, present in this slurry tank will actually have deadly effects on the sensitive bacteria. That is not what I'm including here, but needs to be included. The other thing I'm not talking about is selection pressure. Actually, the deadly effect that antimicrobials will have talks about selection pressure, has a big role to play in selection pressure. The other thing I'm not talking about is that there are different kinds of bacteria who will have different kinds of responses. Not all resistant bacteria are the same. Not all sensitive bacteria are the same. We might have 1000 pathogens that are resistant. We might have 100 different strains or 1000 different strains of pathogens and other bacteria in the slurry tank that are sensitive, and the K value for each of them would be very different. So now notice here, we are not just talking about one bacteria. So if I say we have N is equal to, N is for resistant bacteria, N is equal to 1000 different kinds of bacterial strains in the slurry tank that are resistant at any given time in the slurry tank. Now N is for R, so let me put R here. And for sensitive, let's say I'm using the alphabet K, and we have another 1000 strains of sensitive bacteria in the slurry tank, which can acquire resistance from the 1000 types of resistant bacteria. So for each of them, I need to write a different equation. dRn by dt is equal to Kn Rn Sk. And you can see we'll have multiple combinations of these. The sensitive bacteria, different kind of sensitive bacteria can acquire resistances from different kinds of resistant bacteria. So we'll have a actually humongous model on our hand. So what the authors decided pretty wisely was that let us look at how people tend to model when we have such microbial diversity. One method is take one antibiotic. So let's say I chose ciprofloxacin, which is commonly used in our country, and I take one pathogen. Let's say I choose Escherichia coli or E. coli, and I'm looking at one particular kind of gene that will be transferred, horizontal gene transfer, and I'm looking at one particular gene, let's say PARC. PARC is a gene that will confer antibiotic resistance in certain kinds of bacteria against ciprofloxacin. Alrighty, in this case, life becomes much easier, the model becomes easier because we have one pathogen, one kind of resistant and sensitive strain, we have one kind of antibiotic we are looking at. So, as I mentioned, the resistance against an antibiotic can be very specific, can be, not always, and we are looking at one kind of gene, so one kind of transfer constant. Now, these of course have limitations because slurry tank or environmental matrices are very complex. So, the smart thing the authors did was they were like, we are going to find a midway between taking one of each kind and assuming what is applicable when we have one antibiotic, one pathogen, one gene in a slurry tank, versus when we have millions of different sensitive and resistant bacterial strain in a slurry tank and a cocktail of antibiotics and plethora of antimicrobial resistant genes in a slurry tank that can be transferred from resistant to sensitive bacteria. So the mid ground that they found was that we can say that we have three different kinds of antibiotic against whom the bacteria will have antimicrobial resistance. The first was bacteriolytic. These concepts are important to understand to write the empirical model, so don't get lost, just stay with me here and we'll get to our empirical model. The other thing is, once you get into the modeling business, you need to learn how to very quickly grasp the fundamentals of a new field, unfamiliar field, and then write the models and collaborate with people in that field. So one is bacteriolytic, the other is bacteriostatic. And the third is multi-drug resistance, NDR. So bacteriolytic is when an antimicrobial will kill the cell, cause the cell to disintegrate, to undergo cell lysis, which is the cell is breaking down. Bacteriostatic is cell lysis. Don't worry, this is not an exam. I'm not going to ask you what cell lysis is. But I need you to understand how we are going to use the principles I'm teaching you here to write the model. Bacteriostatic is when we are stopping the growth. The microbial, antimicrobial is able to stop the growth of new bacterial cell. And MDR is a plethora of antimicrobials that can kill or not kill the bacteria or the microbes. So this said, we are going to look at 3 different kinds of antimicrobials. Broadly, bacteriolytic, bacteriostatic, and MDR. There are nuances here. There are some antibiotic that may be bacteriolytic when used individually, but when used in cocktail or in combination with other antimicrobials, they can have bacteriostatic or both effects. And then it is quite possible that some bacteria develops multi-drug resistance, so they are resistant to bacteriolytic antibiotics, antimicrobials, and also bacteriostatic antimicrobials. Okay, so remember our very basic equation that we had on this slide. This becomes slightly complicated, not really complicated, but we have 3 of these equations now. What we have is bacteria that is resistant, let's call bacteriolytic, use alphabet L. For static, I'll use small s, and for MD, let's use U. Okay, LSU. So, let's say a bacteria that is resistant to an antimicrobial, which is bacteriolytic. So, L for bacteriolytic, it interacts with a sensitive bacteria, and now we have 2 bacteria that are resistant to this bacteriolytic antimicrobial. And let's say the rate constant here is K beta L, right? K was the rate constant in the previous slide too, but here I have added beta L to show bacteriolytic. Now we have another equation for bacteriostatic. And then for multi-drug resistance. Now for each of these, we'll have rate constant. So basically, for each of these, I can write the rate at which RL is changing with time, RS is changing with time, and RU is changing with time. Practically, the rate at which resistant bacteria is changing with time is what I'm interested in. That's another assumption that was made in the study, which will be equal to rate at which RL, RS, and RU are changing with time, which comes out to be S so basically the rate equation that we had here, just add them all up. Okay, so this is the rate at which the resistant bacteria are changing with time because of horizontal gene transfer between resistant and sensitive bacteria. This is another thing that's also happening, which is the resistant and sensitive bacteria are growing and dying. So we need a growth term and a dying term. So let me just give you an overview. We are going to find out rate at which resistant bacteria are changing with time, the rate at which sensitive bacteria are changing with time. We are also going to look at the rate at which antibiotic concentrations are changing with time. More on that later. So dr by dt, the rate at which resistant bacteria are changing with time, will include a growth term, will include a natural death term, should include an influent term, because remember, we are talking about a CSTR where there is an influent of resistant bacteria, sensitive bacteria, antimicrobials, and all the other nutrients coming in, minus the effluent, what is leaving the slurry tank, plus minus any decay, any reaction happening inside the tank. This is your mass balance. This is how we are going to do it. So we have, and the reaction plus minus decay will include horizontal gene transfer, which is how a resistant bacteria converts a sensitive bacteria to resistant bacteria. So we already found this term is actually the part of the reaction, the accumulation or decay happening in the reaction tank. But we also need to look at the growth, death, influent, and effluent. So what does the growth look like? Now we have a fixed slurry tank. It has nutrients, and slurry tanks tend to be very rich microbially, so very high number of bacteria. And the authors decided that logistic growth was the way to go, and I think it was a very smart choice. But there is a difference here. Logistic growth assumes that there is binary fission of microbes happening, which will happen in case of the pathogens that they selected, which are bacterial. However, logistic growth says that the only issue that is stopping the microbes from reaching nearly infinite number in a finite amount of time is that the nutrients are limited, the tank is limited, the maximum volume in the tank is limited, which is true in this case. However, there's another very important limitation, which is the presence of antimicrobials. Basically, we have bacteriotoxins present in this tank which are going to impede the growth of the bacteria. So, our logistic model has to be modified. Thankfully for us, in multiple studies, people have figured out what kind of modification works well when we are adding antimicrobials in the mix. And the modified logistic model that we are going to use, that the authors used, and which typically we use, is modified logistic model with Hill function. And I will show you what this looks like in a bit. So now the growth model for both resistant and sensitive bacteria is here. We have a very simple death model. Death model is going to assume that this is natural death. This is not because of presence of antimicrobials, this is not because of competition or lack of resources, which is included in the modified logistic model that has Hill function in it. Influent will depend upon two things. First, what is the current volume of the slurry tank? Now, in UK dairy farms and also across the globe, the slurry tanks, when they are empty, there is still some volume that remains, volume of slurry that remains in the slurry tank. There's always some amount of slurry that remains in the slurry tank. So there's always a background V1 or V initial which is present, and then there is Q in or volume increase that happens because of the flow coming into the slurry tank, and then there is a Q out, which is decrease in volume happening because of the removal of slurry from the slurry tank. And now here's the thing, when the water comes in or water goes out, it's going to dilute, increase or keep the concentration of microbes, resistant or sensitive, constant. So what we need is we need a term for influent, and I'm going to show you in the next page how that will look like. Effluent also, and decay reaction, we have already taken a look at it. Okay, before we go into the volume, let's look at some of the assumptions that people made in this study. It's a very good study and I encourage you to read it, especially if you're interested in microbial rich environments such as WSPs, ASP, any kind of wastewater treatment, any kind of cleaning up of rivers that are heavily contaminated, or even contaminated soils where you want to use bioremediation. Not for the microbial part, but for the mathematical parts. Very simple mathematics. Nothing that we have not studied so far. Okay, the first assumption, as I said earlier in the class, was that they assumed that this was a CSTR, and they assumed that it was homogeneous, well mixed, which is important. It's continuously stirred tank reactor, which may or may not be the case in actual life, but that was a decent assumption to have. Now, because of this assumption, the bacterial concentration, nutrient concentration, the antimicrobial concentration will remain constant throughout the tank. The other thing they assumed was that the tank is open, which is very important. It's open to the air. Of course, there's influent coming in, there's effluent leaving the tank, so it is open. It's not a closed tank, it's not a batch reactor, but it is open in the sense that it is in touch with the air, which means there's continuous oxygenation happening, so the DO is not falling down because of any excessive microbial activity. And because the tank is open, now we are not talking about theoretical model, right? This is a model for actual dairy farms. So because it's open, in case of rain, dilution will happen of the slurry. But again, they assume that that dilution would be negligible on because the tank is huge and they were not expecting crazy amount of rain. The other assumption is that the inflow is constant, which is not always true in case of a dairy farm, which is also not true in case of a wastewater treatment plant. Typically, there are hourly, daily, even in this case hourly variations in the influent coming into the slurry tank, but to make model a little easier, they assume it's going to be constant. And I think for the purpose of the model, this was a pretty good assumption. It may not be valid in certain other cases when the objective changes, but in this particular case, it was a pretty good assumption. The next thing they said was that, remember I told you that there are three different kinds of antibiotics assumed in this study. One are bacteriolytic, one are bacteriostatic, and the other are multi-drug. So basically, they are broad range. Bacteriolytic, they kill the cell by breaking it apart. Static, they stop the growth. Now, in this study, they assume that these are the only three kinds of antimicrobials that are used in dairy farms that will be found in the slurry tank of the dairy farm, which may or may not be the case, because let us look at it nicely. There are some, as I mentioned, there are some antimicrobials that belong to one category, but in presence of other chemicals, other antimicrobials, they start behaving like the other category of antimicrobial, but they assume that's not the typical behavior, so they assume that's not happening. Now let's come back here. So death here is natural death. Growth is modified logistic model with hill function. Influent is because of the constant influent coming into the slurry tank. Then there's the effluent, and the reaction we already modeled is because of horizontal gene transfer. Okay. Now let us look at the slurry tank. Now, the slurry tank, as I mentioned, the volume of the actual tank remains constant, but because of the constant influent coming in and then the tank being occasionally cleaned out, the actual volume of the slurry in the slurry tank keeps on changing. So, that can be modeled like so. Vt is the volume of the slurry tank at any given time t is equal to V1 or Vi, which is the initial volume of the slurry tank. Remember, I told you even when the slurry tank is cleaned up, there is always some volume of slurry left inside it, plus minus, okay, what are these two functions? This function At is basically influent, slurry moving inside the tank, and omega t is the slurry moving out of the tank. We need to model what At and omega t look like. Now, as slurry moves into the tank, the concentration of antibiotics, the concentration of the different kinds of bacteria, resistant or sensitive, when we are looking at concentration of antibiotics and bacteria, we need to account for the volume term. The volume term needs to be included in it, and that will look like this. The concentration of antibiotics or bacteria or whatever we are talking about will vary with how the influent is changing with time, how the volume is changing with time. This needs to be included. This particular term needs to be included. Okay, this is the rate at which the volume is changing. So, if we derive this, we will get dV by dt, which will tell us how the volume is changing with time, and any given time this is the volume of the tank. Now, as I mentioned, with the concentration of antimicrobials and nutrients will also be impacted by volume. This is how we are going to include that. Briefly, the concentration of any term that we are looking at, dCt by dt, it will depend upon the influent coming in, the effluent, the concentration leaving, and any kind of reaction that is happening. So, we need to account for each of these three, and in the next lecture I will tell you what they look like. This is all for today's lecture. We will finish this model in the next lecture. See you there. Thanks.